Thanks for the introduction. It's so nice to see so many familiar faces and names here. So yeah, I guess I'll just dive straight into it. Just a little bit about me. I'm doing my PhD now, actually entering my third year at the University of Glasgow. But before then, I was with Marine Scotland Science in the Renewables Advice Department. And way back when, I also did my undergrad in marine biology at the University of Aberdeen. And here is me cutting up some samples for my PhD research. My wider PhD topic is on utilizing fatty acids and stable isotopes in order to enhance our understanding of cetacean ecophysiology, as well as exploring their applications in understanding factors of marine mammal stranding events. So I'm with SMASS, which is short for the Scottish Marine Animal Stranding Scheme. It's the dedicated research and reporting scheme for stranded marine animals in Scotland. And we've been doing stranding surveillance since 1992, so we've been going for about 30 years. We have a very dedicated volunteer network as well as some permanent staff. We do some data collection, carcass collection, as well as collect samples for all kinds of research. We normally say that strandings data is dead useful, and this is because the information that we do collect from strandings support research projects that can inform us about health and disease, migration, behavior, but also things like pollution, population structure, and that's just naming a few. Now, there are some biases with that, but because cetaceans are so difficult to study in the wild, this is kind of close to the best that we got, especially for things like health. So a percentage of the cetaceans that we do get in, we do a necropsy on. That's essentially another term for a post-mortem. We follow a European standardized protocol. We gather things like age, sex, nutritional condition of the animal, as well as some morphometric data, so weight, length, and girth. We also do gross pathology on all inner organs, and altogether we're able to often, most of the times, determine the status of an individual at the time of stranding, as well as the cause of death. So really helpful. And of course, we've amassed a large number of samples. We have about 30 years of samples stored at different temperatures, so -20, -80, as well as some in formalin and alcohol. So we have about 3,000 samples in our archive, and if you have any project in mind, please do not hesitate to get in touch. We'd love to hear from you. So why do we go through all this effort? Why should we, even bother collecting samples at all? I can think of three very important reasons for that. First of all, they're very charismatic species. Everyone loves whales and dolphins. So because of this, we call them umbrella species. So by protecting them, we essentially indirectly protect other animals in the ecosystem. They're also apex predators, so they're top of the food chain. They accumulate a lot of things in their bodies, so they're considered environmental sentinels. And by studying them, we can possibly find out more what is happening, in the ocean as a whole. And finally, and probably most importantly, it is the law. So we have to, by law, monitor the abundance, the distribution, the pressures and the threats that these marine mammals experience in order to protect them better and assessing and maintaining a favorable conservation status. So my research is on isotopes, and I know this is the Biogeochemistry Forum, but I thought we just kind of return to the textbook definition of what is an isotope. So an isotope is an atom with the same number of protons, but a different number of neutrons. And what we call an isotopic signature is the set of ratios between the isotopes of an element in a sample. So that can be, carbon, nitrogen, sulfur, oxygen, you name it. And these are incorporated and stored in the tissues through the diet on different timescales. And just like fingerprints, we can use them to track and trace throughout the ecosystem. So I've included two figures here that I find are really helpful in understanding the concept. The circle up here is what we call turnover rate, so the rate that the elements incorporate into the tissues of an organism, and particularly here in cetaceans. You'll see at the top of the screen there, we have days to weeks, so tissues with a very rapid turnover rate. Can be liver, but can also be things like blood plasma. And if you turn all around the circle, you see there's no turnover rate in things like baleen and teeth, which is really useful if you want to find out the lifetime stable isotopes of an organism. And if you move towards the bottom of the screen, we have something called an isoscape. That's essentially an isotope map. And it's an example here of how the UK Shelf Sea can be divided into these 5 estimated discrete regions of similar carbon, nitrogen, and sulfur isotope ratios. And that's really useful in marine mammal research, looking at things like migration and movement. So of course, we use isotopes in marine mammal ecology, thanks to the integration of the elements from the diet into the different tissues. So different studies, there are lots of them, use isotopic compositions to study things like trophic ecology, but also diet and habitat use. And I've included a figure here from one of my colleagues and a SMASS member, Tessa Plint, and she used stable isotope ecology and SMASS samples on looking at interspecific dietary overlap amongst dolphins that are in the Northeast Atlantic. So I can send the paper in the chat, but essentially the different colors are different species of dolphins, and where the ellipses overlap, that means that the species overlap in their dietary niche, and when they don't overlap, they obviously don't. And that's really useful to know where they're moving and what they're feeding on. So if you're a stable isotope ecologist, you probably come across this term, you are what and where you eat. So do keep this in mind for the second half of the presentation. So my thinking went that when the cetacean is alive and it's roaming the oceans, it's incorporating all of these different elements into its tissues when it's moving and feeding. If it ends up here, and not all cetaceans do, but if it ends up on the beach and we're able to get to it and take some samples or even a necropsy, there's huge potentials in using stable isotopes to understand at least part of the life history patterns of the animal that is now dead. So I'm using a case study of a mass stranding that happened last year on the Isle of Lewis. You might have seen this in the news. We had 55 long-finned pilot whales wash up in July of 2023. There was a very, very quick response from different volunteers, but also organizations and the general community. We got almost all of them post-mortemed and all of them sampled. And there's, obviously this is a very traumatic event and people want to know why it happened, what factors can be behind it. So there's a very strong social and political drive that's kind of pushing this investigation forward. Some interesting facts about this is this animal, these species, haven't commonly been seen around the area. So we have sightings data spanning about 30 years, and they're not a common species encountered. They were also in quite good body condition. However, there was no evidence of recent feeding, so most of the stomachs were actually empty. And some females were actually pregnant at recent stages. But I would say the main question here, at least for me, is why was this deep pelagic species inshore before it stranded? And if you keep up with the news, you might have seen this happen again. This time in Orkney, there was 77 pilot whales stranded once again. So it's very relevant right now as well, just in that investigation. Just a little bit about the actual species. The long-finned pilot whale is a frequent deep-diving oceanic dolphinid, frequent in the North Atlantic. It's very social creatures. They live in these matrilinear pods, but behaviorally, it's quite interesting because they aggregate when there's any disturbance surrounding them. And this social nature of the species could be a blessing, but also could be a curse because they're considered the most commonly stranded cetacean worldwide. And just returning back to them the question, what was this deep pelagic species doing inshore before it stranded? So I mentioned there was an investigation about this. We're looking at things like proteomics, population genetics, metagenomics, reproduction from different institutions around the UK. And stable isotope analysis plays a part in that investigation, particularly because we want to understand more about their foraging history before they stranded, and this is something unique to stable isotope analysis. So essentially, if you remember that I said you are what and where you eat, we can use stable isotope analysis to hopefully find out more about the foraging ecology of the animals leading up to the stranding and complement those other investigations that are ongoing. You might also remember this figure of the different turnover rate of the different tissues. So I'm using this principle in the final chapters of my PhD. For stable isotope analysis, I'm applying carbon, nitrogen, and sulfur stable isotopes to two different kinds of tissues with two very different turnover rates. So I'm looking at liver, which has a very quick turnover rate, and muscle, which has a slower turnover rate of the individuals stranded. And by doing this, I can hopefully be able to reconstruct the recent diet and movement of these animals and essentially see if there were any changes on these different timescales. I also hope to be able to look if there were any inter-individual differences between groups. So maybe there were differences in foraging ecology for females versus males or for adults versus juveniles. And this will kind of help rule out any factors for the mass stranding. And my colleague is also doing skin and blubber on the same animals. So towards the end of it, we'll have a whole kind of profile of each individual and hopefully their different foraging ecology and movement patterns. So what's left for me to do? Wait for my data, but also then once I get the data, I'll need to apply some stats on it. So Bayesian mixing models for diet reconstruction, but also movement using isoscape models. So kind of my call to you all is if you have any isotopic prey data from the region, or if you just have any experience of these methods, I would love to hear from you. So please get in touch. My email's here at the bottom. So kind of rounding it off, what are my hopes, my dreams, and my goals with this part of my PhD? I want to be able to showcase the potential of using stable isotope analysis in standard strandings protocol. I also want to add another depth of knowledge to these species because essentially they probably are native to the UK, but we don't know that much about them. The population might be changing, so we do need to understand and kind of have a finger on the pulse, a baseline on where they're moving, what they're eating, why they're in the area that they are. And by using stable isotope, we can add to that knowledge, but we can also look at things like fisheries overlap. Are they maybe feeding on the same fish that are commercially interesting, or potentially how they're moving in and out of different MPAs? So there's lots and lots of different strands to this research and lots of potential. So yeah, that's kind of a whiz through of my PhD so far, but happy to take any questions.
Thank you. So once again, we are now able to take any questions. You're getting a lot of applause from the audience. If you'd like to ask a question, you can write it in the comments, or you can unmute yourself, put your hand up, whichever you'd like to do. So this is, I think I'll start with the first. Oh, we've got one hand raised up, so we'll go straight to Chris.
Hi, folks. Can you hear me okay? First thing.
Yes.
My mic's sometimes a bit dodgy in the office. Anna, thanks for a great talk. Well, we've talked before. Looks really interesting. I'm going to give you a question about. So you've got all these samples, and you're going to compare the liver and the muscle. One thing I would say, I'd just be recently talking because you're going to do sulfur as well. So one of the differences you're going to have between those tissues is the amount of lipid that they have in them. Okay. And it's something that us isotope people always go on about and we obsess about and we talk about too much. But one thing that we've noticed is that the lipid extraction affects sulfur. Okay, so sulfur is a really weird isotope. It's really complicated. But so
I've heard.
You'd have to split samples for carbon and nitrogen probably anyway, because lipid extraction can affect your nitrogen. So think about that with your sulfur as well, just to warn you now, yeah?
Thank you. I have lipid extracted as well, but I'll get in touch with you and we maybe chat offline.
Okay. Yeah. So I'll let everyone else ask questions now.
So the question I have is, so you've mentioned that there was the two strandings, and it might well be very early days, but were there any similarities or trends that have been identified yet as to what might link those strandings? Is there anything that your work has found so far, or is it again too early days?
I think it is kind of early days, especially for the more recent stranding, and because I'm so heavily focused on the earlier stranding, I think in future it would be quite interesting to compare the two. But for both kind of strandings, one similarity was they were in good body condition. Maybe we're thinking there one might have been a mating aggregation between multiple pods. So we are doing genetics work on both of them to see why they were in that area. But Orkney and Isle of Lewis are kind of dissimilar areas, but still, general speaking, same area. But could be an interesting other PhD.
If you need another one. And I guess as well, so how susceptible are pilot whales to their marine environment? So, going back to Marta's talk with the nutrition cycle, and I know that's based in the Arctic, not based around our oceans, but how, I mean, and maybe this is a question that Marta wants to come in and ask as well, but would things like this, could these things be like this be linked to these strandings, the just general kind of the change of ocean chemistry? I'm not too familiar with pilot whales. I'm not sure how easily affected they are by their environments.
I think in terms of geochemistry, I think Martin's best placed to answer that. But they are, generally speaking, susceptible to different sort of disturbances. But we don't know, again, if there was a disturbance that moved them there, or if maybe they were following prey up to warmer waters, things like that. Maybe they weren't familiar with the topography of the ocean area as well. A really interesting point, actually.
I think, Kieran, you had your hand up next.
Thanks, and really a couple really good talks, actually, very interesting. Question for Anna, and I know we've chatted before a bit about your project and also the other work that we've been doing at CERC, looking at stable isotopes in cetacean teeth. Thinking about you put out the call for information on stable isotopes for prey species for pilot whales. So I know that cephalopods would be a predominant prey. Are gadoid fish? I don't really know much about these whales actually, or these dolphins, but would gadoids like cod, haddock, whiting, could they potentially be part of the diet for these?
Essentially, we do see evidence that they follow, I know they're not gadoid fish, but mackerel as well. I mean, gadoids could be useful. I will say that for the 2024 stranding that I'm not investigating, is we did find shark purses in the stomachs, which I haven't really encountered in any of the literature. Yeah, so
that's interesting.
We don't know.
Yeah, okay. One to keep in mind, so we have some samples here, which I'll hopefully be running with Rona soon, and we'll have for whiting, hopefully in the not too distant future, some data for whiting, but that may be extended before the end of your PhD anyway, to other gadoid species anyway. So, but yeah, definitely keep in touch. And also, if anything, obviously it doesn't help in terms of the stranding for information, but thinking about the teeth as well and the kind of long-term information that we can get out of that. So, but I'll definitely catch back up with you and Andrew Brown with the data that we've got.
I'll come see you.
That would be great.
Thank you very much. Chris, you have another question?
Yeah, if I can be rude enough. Anna, these are from the Isle of Lewis, yeah? You have to forgive me, I'm a blow-in, I'm a recent person in Scotland, but that seems to be right on the edge of all the borders in your isoscape. That's going to complicate things. I mean, how far do these things travel? Have they got like a home range of, don't know, 20 kilometers, 2,000, whatever? I don't know. How far do they go?
We don't really know in this particular bit of the ocean, right? That's why we're trying to find out.
Okay.
And I really want to know where I mean, my thinking goes like this, and do correct me if I'm wrong, but we will probably still be able to see if there was a change in sulfur, but we won't be able to pinpoint that, right?
I mean, you may see variation or switches, right. Yeah, but it's just if you're trying to reconstruct where they've been.
That's the idea.
The other thing is how steady these isoscapes are in time, because you might have a really stormy year versus a not very stormy year. So it's all part of the fun, it's all part of the enjoyment of doing this kind of stuff, is trying to work it out. But I'm just, you're going to get data and you're going to get interesting data. It's just trying to reconstruct exactly, because it's easier when they're up on the Orkneys or something, because it seems to be in one kind of area of your isoscape. But it'd be interesting. It'd be interesting to see.
So a visit to Scene coming up.
Come and see us. Well, or we could go down we could meet at CERC and all get together. It might be a good idea. Yeah.
Thanks.
Cool. Cheers. Thank you.
I love how collaborations are already being formed and
I need all
lifelong
the help.
partnerships. We have time for one more question. Rona Sinclair, you have your hand up.
Hello. Yeah, thank you both very much for the talks. It was really, really interesting. I have a question for you, Anna, if that's okay. It's kind of building on what Chris was just talking about there. And I've never seen this the coloured map before, or I don't know much about stable isotopes. I was wondering whether you could just describe a bit about what the different types of samples, so like the skin samples all the way around to like the baleen samples that you could use to understand. Obviously, they have different time frames that you can use those samples for to understand potentially where the animals might be moving. But what do the different colours on that map mean? Is that relating to the different like carbon, nitrogen, sulfur?
Yeah.
Sorry for the very ignorant question.
No, that's a really good question, actually. Sorry if I might not have explained it well enough. Essentially, I took this from a report on fisheries, so pinpointing where fish move. So the different colors are different values, kind of shared values of carbon, nitrogen, and sulfur. And they all tell us different things about the environment. So potentially, how close is it to shore versus how far it is, or depth layers. I could be wrong in kind of saying that, but I see some nodding encouragement heads here, so hopefully good. And you mentioned about the skin and the different tissues. I guess you were referring to this figure.
Yes, and I was just wondering whether there's, I was actually thinking that they were kind of related in those figures, where the long, like the colors might have related to the different kind of.
Not really.
Okay.
You can use the
Sorry.
No, that's actually a really valid thought process, but they incorporate on different times in the different tissues.
Okay.
So my principle is that because they incorporate different times, you can use the isoscape map for that. But it's not like these particular tissues correlate to a point on that map. That makes sense.
No, okay. Yeah, no. That does make sense. Yeah, I was just wondering, I suppose, where my thought process is going was if you were able to look at, for example, the no turnover time, the baleen or the teeth, where whether you could see whether the whales were coming from down in like off the southwest of England, for example, and that related to then one of these kind of carbon, nitrogen, sulfur, and if you picked one of those up. Anyway, this is my total ignorance of how stable isotopes work, but it's just got me thinking, and it's very interesting.
Yeah, no, really good. Maybe something for the future as well.
Yeah, thank you.